Hi, I'm Dr. Amanda Haage. I'm an assistant professor here at the University of North Dakota in the Department of Biomedical Sciences, and I am a PI on the cellular pillar part of NDACEs. So what I work on is how cells in the body move through the body. So there's a lot of different important contexts for why your cells may move in their body. A lot of the cells, when we are developing, they don't start where they kind of need to end up. So when you're creating an organism, a lot of these cells have to move. But then one of the more important contexts to long-term human health in which cells move is cancer metastasis. So that's the process where you have cancer cells and tumors that start in one place, say in like a primary tumor, and then the cancer cells will migrate or move through the body to a secondary location to create a secondary tumor or multiple tumors. It's one of the biggest health concerns that we face kind of in the cancer field in general, because it comes once those cancer cells start to spread, it becomes really, really hard to treat and find them all and those kind of things. So I look at this process of how cells move, particularly in how the environment around the cells and all the different conditions in the body affect their ability to move. And my role here with NDACEs and my work specifically on this project really gets at that process specific to breast cancer cells. So most cancer cell types metastasize or move through the body to do that process of creating secondary tumors. Breast cancer cells are no different. So my role here is to use a lot of these environmental conditions, specifically these scaffolds that have been created by my other colleagues on the program that are designed to mimic different environments in the body. And so then I study how those cancer cells then will move through those scaffold mimics, so we get an understanding of what that may look like in the body and how those cells interact with their environment, how they sense everything around them, what chemicals around them, the physical properties of what's around them, and then how that affects how they move, when they move, those kind of things, and creating those secondary tumor process for things. So one of the really nice things about this project is that it's incredibly student-friendly in creating projects for students, because there's all these different environmental conditions in the bodies. Our bodies are super, super complex. So we have all these different types of proteins that are around. You have different stiffnesses, from like your bone being a very stiff environment to your brain being a softer environment. And then all of these chemical signals, your kind of growth factors, or anything kind of floating around in the blood that's going to, tell your cells to go someplace, do something, those kind of things. We have all these different conditions that make nice little projects for students to take one of those conditions and really, become the little expert on that specific environmental condition. So we have a wide range of projects that students would be able to complete on those different environments, testing how cancer cells move in underneath those conditions for those kind of things. And one of the greatest things I would say, as far as why you should work on this, is what I found when I started my career in science is that the most fascinating thing to me was being able to actually see those cells move. So we use a lot of time-lapse microscopy techniques, so we actually get to use video recordings on a very, very tiny scale and actually see those cells move in those environments. And it really helps you connect maybe the things you're learning about cell biology, cell proliferation, organelles, your mitochondria, your cytoskeleton, all those little itty-bitty things that you're learning about and you see pictures of, but you don't actually see live happening, because we can't without the aid of microscopy. But doing time-lapse microscopy, then you get to actually see that stuff and connect it to this macro scale, connect it to these things that are happening to real people, affecting their real lives, like cancer. And then the other pitch that I have for you is coming to UND for your pre-health education. So I'm also the creator and primary instructor for our new anatomy and physiology sequence of courses here. And one of the best things that I love about teaching here is the fact that we have a cadaver lab on campus, and we have the ability to get undergraduates involved in that cadaver lab. It's been one of the most fun things I'm doing over the summers. I have my students that are going to be TAs now for this course, going through and getting some hands-on dissection experience, but also even watching students come through the cadaver lab again, just like seeing the cells underneath a microscope. You can see the real body, and so instead of looking at all these pictures of muscles and bones and stuff, you can see and interact with the real thing. And one of the things I'm also trying to do with these new courses is teach them through a diverse lens. So there's a lot of diversity topics incorporated into the courses because, you know, how our anatomy and physiology are different from one another and how we have this variation across the human population affects how we move through the world. And it's going to affect the patients that you're going to treat eventually, and hopefully giving you the experience to become a healthcare professional on a more global scale in a diverse world.